Couple more questions from chapter 6. How do concentration and inhibitors impact enzyme function? So we left off on Friday basically talking about enzymes working as biological catalysts. And biological catalysts, what they do is they speed up the rate of reaction. And some of you, I think, got a little bit confused by this on your quizzes. And you either didn't talk about that enzymes speed up the rate of reaction, so even exergonic reactions require enzymes because they have to happen at a rate that's necessary for living tissues, okay? Some of you didn't mention that and so you lost points, and then some of you, I think, got confused about activation energy and what that actually means. Just because there's an activation energy does not mean that an exergonic reaction requires energy, okay? The activation energy is basically just what's required to get the materials to come together and to get the kind of strain on bonds necessary for the reaction to happen, okay? And so activation energy, if apart from a catalyst, is basically like at what rate do these things need to be moving to collide with one another? Right? Because if they're going to react, they need to come into physical contact. And so what enzymes do is make it more likely that they're going to come into physical contact, but still they have to be moving, right? So if you're at, you all know the temperature at which all molecular motion ceases? It's called absolute zero. It's negative 273.15 degrees Celsius, okay? Which I don't know what that is in Fahrenheit. I don't want to do the math. And so, or 0 Kelvin. And so at that point, an enzyme's not going to do any good because nothing's moving. They're not going to come into contact, okay? But what lowering the activation energy does not mean is it's not going to make a reaction more spontaneous. Some of you wrote that, and that is certainly not what it means. Enzymes do not change the delta G, so they're not going to change the spontaneity. If it's spontaneous, it's going to be spontaneous regardless of whether or not an enzyme is present. What an enzyme's going to do is shorten the amount of time you have to wait for the reaction to happen. Okay, doesn't change the thermodynamics of the reaction itself. All right, so now even though enzymes are not a reactant, they can become saturated. So they're not used up by the reaction, but they are facilitating the reaction. And there are only certain numbers of enzymes to actually act as catalysts. And what if they're all full, right? If they're all working on reactants, then you've got reactants that are ready to react, but there's nothing to help them to find each other, right? Does that make sense? So the enzymes can become saturated. And so enzyme concentration correlates in a linear way with reaction rate. The more enzyme you have, the faster your reaction rate, and it's a linear relationship. If you have twice as much enzyme, you're going to have twice as much or twice as fast a reaction, typically. If you've got 3 times as much enzyme, you're going to have your reaction rate happen 3 times as fast. It's a nice linear relationship between your enzyme concentration and your reaction rate if everything else is constant, right? You're only changing the enzyme concentration. Now, because the substrate is a reactant, it's being used up. It's being used up by the reaction, meaning it's disappearing. And if it's a different enzyme that catalyzes the reverse reaction, then I mean, that reaction is in essence, without that second enzyme, it's not reversible. And so your substrate, once it becomes product, it's product. Okay, because remember, if you've got an exergonic reaction, the reverse reaction is going to be what? Endergonic. So if you have an exergonic reaction where your products have less free energy than your reactants and you're releasing energy, the reverse of that reaction is going to be endergonic, where the products have more free energy than the reactants, and now you really need to provide some energy to fuel that process. Okay? And so substrate's going to be used up. And so substrate concentration actually has a logarithmic relationship with the rate of the reaction, with everything else being constant. And so your substrate concentration, as you put more of it, it's going to speed up your rate of reaction, but it's going to get to a point at which it doesn't really increase it much anymore. You get to some threshold, a maximum reaction rate. A maximum reaction rate. And this is because you're holding your enzyme constant. And so you can put more substrate in, but you only have a certain amount of enzyme to facilitate all these reactions. Okay? And so as substrate concentration goes up, your reaction rate goes up. As enzyme concentration goes up, your reaction rate goes up. Okay, but the exact relationships are a little bit different. Okay, now we need to start talking about inhibiting enzymes. So enzymes can be inhibited by molecules that are called inhibitors. And there's basically two forms of inhibition. There's competitive inhibition and there's non-competitive inhibition. And when you see the word competitive, what do you think of? Do you think competition? Think it's something, two teams maybe competing for the same goal, right? And that is to win, right? And so in competitive inhibition, you have two substrates competing for the same active site on the enzyme. Okay? Two substrates competing for the same active site. In non-competitive inhibition, the two substrates aren't, the inhibitor isn't competing with the substrate for the active site. What it is just it's changing the enzyme. And so non-competitive inhibitors, what they tend to do is inactivate the enzyme altogether. They're not just out competing the substrate for the enzyme, they inactivate the enzyme. They work as an off switch. An off switch, okay? So this is often what we call allosteric regulation. And so what that does is it changes the enzyme's chemistry or changes the enzyme's shape to make it no longer have a high affinity for its substrate. And again, it can act like an off switch. And you're like, this is so cool. Because if I were a cell and I want to be poised and ready to carry out the reactions necessary for my survival, it's kind of nice to be able to have enzymes ready to go but turned off until something is present for them to do. Okay? And then you can turn them on by removing this allosteric inhibitor. Now what you find is for many of these reactions, for many of these long metabolic processes, the final endpoint, the final end product acts as an inhibitor on one of the enzymes in the process. And so we'll see this on Wednesday. On Wednesday we'll talk about glycolysis. And just a little bit of a foreshadow, glycolysis is the process of taking a sugar like glucose and oxidizing some of the carbons to generate ATP and to free up some electrons. Okay? That's what's happening. The end product of glycolysis is pyruvate, and pyruvate acts as an inhibitor for one of the enzymes in the process of glycolysis. So if there's a whole bunch of pyruvate building up in the cell, or a whole bunch of even ATP building up in the cell, it'll stop breaking down glucose and stop generating more. Yeah?
How do you think cases where the inhibitors just permanently shut down the enzyme?
Oh yeah, absolutely. Yep. Yep, absolutely. And to where it's irreversible. Yeah, because some of these inhibitors act as what's called a tag for destruction. And so there's a protein that's called ubiquitin that tags proteins for destruction. And if it's acting on an enzyme, you could call it an inhibitor. And if it tags an enzyme for destruction, it'll basically mark it for it to go into a complex called ubiquinone, and it'll just chew it all up and recycle the amino acids. Yeah. Oh yeah. Okay. Just a couple of key terms. We're going to see a lot of enzymes that are called kinases. Kinases are enzymes that phosphorylate materials. So some kinases will phosphorylate enzymes and will turn them off or turn them on. And then there are enzymes that are called phosphatases, and these remove phosphate groups. So kinases add phosphate groups, phosphatases remove phosphate groups, and oftentimes these phosphate groups act as an on-off switch. Okay? So for some enzymes, you turn them off by adding a phosphate group. Some enzymes, you turn them off by removing a phosphate group. Okay? And so it really varies from enzyme to enzyme, but the general rule is that to phosphorylate something is to activate it. Okay? Is to turn it on. That's not true for all enzymes. Some of them, you actually turn them on by removing a phosphate group. But typically, you turn an enzyme on by adding a phosphate group. And where do you think the source of that phosphate group is? Where does this kinase get its phosphate group to phosphorylate things with? ATP. Because there's a bunch of it, right? Making and breaking 100 million of these every second. 100 pounds of them every day in every cell in your body. There's lots of phosphate. Lots and lots of phosphate. Okay, so here's enzyme concentration. Again, a nice linear relationship. As enzyme concentration goes up, so does the rate of the reaction in a linear relationship. Here's substrate concentration, a logarithmic relationship. And so as you increase substrate, your rate of reaction increases up to a certain point that it can't pass, and that's the saturation level, where all of your enzymes, they're busy, right? The enzymes are the gatekeepers. They're the ones basically controlling how fast the reaction can go. You're like, well, what about if we increase both? We increase both substrate and enzyme. And it's like, you'd increase the rate of the reaction in a complicated way. It'd be kind of both linear and logarithmic. Okay? It'd be like logarithmic where you just keep bumping that bar up of that saturation level. Have you ever seen graphs for carrying capacity? You should have seen these, right? Where it's like there's a maximum number of individuals of a population that the environment can sustain. You seen these? I know you've seen them applied to human populations where like we're getting to a point where we might be reaching the carrying capacity of how many humans this population can, or this globe can sustain. And it's like, well, all you got to do is you kick up the carrying capacity. Just keep putting it up and it doesn't matter. And that's what we've been doing over the last several decades, is just kind of raising the carrying capacity to keep blowing through it. But also, reproductive rates are on the decline in almost every country on Earth. So there's also that helping as well. Oh, also, people in developed countries are waiting longer to have kids, you know, so then it lengthens generation time, so it takes longer to double. Anyways. All right, so here's competitive inhibition. Here we've got a substrate, and look at this. We've got an inhibitor competing with the substrate for the active site. So if it can beat the substrate there, guess what? Reaction doesn't happen, okay? Because this inhibitor's there, and it's binding to the substrate. And we'll see several examples of these, okay, of competitive inhibitors. Here's non-competitive inhibition. Here's the inhibitor. It bound somewhere else, usually called the allosteric site. And what it did is it changed the chemistry and the shape of the enzyme to now it can't bind the substrate. This works more like an off switch, where this works more like when like my wife wants to give me a hug when I get home from work, but all three of my kids are draped over my body, right? There's just no room, right? It's actually kind of fun. It's for a minute, and then you got to get the kids off because they're usually snotty and just gross. Oh man, I'm so terrible. I am so terrible. Okay, so here is we can inhibit something in an allosteric way, but we can also activate an enzyme in an allosteric way. So here's an enzyme that without the activator, it is not the right shape and chemistry to bind the substrate. And then we've got an allosteric activator, binds to that enzyme, changes its shape and chemistry, and now it can bind the substrate. So it actually works both ways. Okay? Now, you can't have a competitive activator. Right? You can't have something activate an enzyme by getting into the binding site, because that's where the substrate belongs. You can only have an inhibitor work competitively, but you can have an activator and an inhibitor work allosterically, non-competitively, changing the shape and the chemistry of the enzyme. And again, a lot of time those activators and those inhibitors are going to be phosphate groups. And here's allosteric inhibition. Okay, we showed that already. Okay, this one's cool. All right, so here we've got a metabolic process that requires 5 reactions. Okay, and each one of these reactions is facilitated by a different enzyme. All right, you see that? So we've got enzyme 1, enzyme 2, enzyme 3, enzyme 4, enzyme 5. And it requires all 5 enzymes to carry out this metabolic process, to carry out these 5 different reactions. Check this out. The end product of reaction 5, so the end of the entire process, acts as a inhibitor on enzyme 1. And so if you build up a lot of this product and it's not being used, it stops the process at the beginning so you don't keep building up all of these intermediates. Isn't that beautiful? And we call this feedback inhibition. Okay? And it's really, really common in metabolic processes that the end product inhibits something along the way. We also have feedback activation, where the end product makes it happen at an accelerated rate. And there are really only a few good examples of that. One is in childbirth, that the hormone released as you put pressure on the wall of the uterus leads to an increase in the strength of contractions. So it leads to an increase in the pressure on the wall of the uterus. And you get what's called feedback activation or feedback enhancement. And the other one is, the other good example that I think about is with lactation as well. Okay, that you get like the sucking force that encourages the glands to release their product, and once they start to release their product, it encourages the glands to release their product more easily. Okay, you get like a feedback enhancement. But we see feedback inhibition all the time, all the time. All right, last question. What other factors regulate enzyme function? What other factors regulate enzyme function? This is going to be a very important question to answer for the lab that we're going to make up the week right before finals. Okay, when we're going to do enzyme activity. Lab this week, we're doing fermentation. We're going to take yeast and we're going to feed them a whole bunch of stuff. We're going to give them sugar. We're going to give them 0-calorie sugars. It'll be fun. I mean, it's kind of like you set them up and then you just kind of wait for a while and see what they do and work on other things. But anyways, it's kind of fun at the same time. All right, well, enzymes tend to have an optimal pH range. Enzymes tend to have an optimal pH range. So low pHs, acidic conditions, and high pHs, basic conditions, they can change the chemistry of amino acids. And if you start to change the chemistry of amino acids, you're going to change the way your protein's folded. Okay? And this is how you can cook protein in an acid. Have you ever had ceviche? Where you cook fish in like a citric acid from lemons. So you're cooking the fish. You're basically accomplishing a similar thing to when you heat it up, right? As you're unfolding proteins and you're cooking it, but you're not using heat. You're using an acid. But it doesn't kill the parasites in the fish, so you're like, is it worth it? And the answer is yes, because it's delicious. As long as you use marine fish. Freshwater fish, probably not worth the risk. All right. Pepsin is an enzyme that works in our stomach in digestion, and it has an optimal pH of 1.5. That's awesome. And so that is a super duper low pH, and that's where it operates most efficiently. Temperature, increases in temperature tend to increase the rate of a reaction. Reaction rate doubles for every 10 degrees Celsius you increase. And this is because the faster things are moving, the more often they come into contact, right? And that's what you need in order to have the reaction. However, high temperatures can break bonds. And what holds the protein in its three-dimensional shape? Hydrogen bonds, mostly, but also some covalent bonds. And so when you start to heat it up, you can break those bonds and unfold the proteins. And that's what you do when you cook a fish using heat. Okay, you heat it up, you break the bonds, it unfolds the proteins, and now you've got a cooked version of that protein. Okay, you can accomplish the same thing with low pHs, but so with enzyme catalyzed reactions, you see this as you increase the temperature, they happen more rapidly until you get to a point where you start to unfold the protein, and then all of a sudden they don't happen at all. And so here you can see that the effects of here's the effects of pH. So here's a typical cellular enzyme with its optimal work at normal body pH. Here's an enzyme that has its optimal at a basic pH. Here's pepsin with its optimal pH about 1.5. And then here you've got the effects of temperature. So as you increase temperature, the reaction rate increases until that enzyme starts to unfold. And so this, with Siamese cats, you get some places where the body's really, really warm, where it messes with the ability to deposit pigment. Okay, and it actually lightens their color. So if you have a Siamese cat or you know anybody that has a Siamese cat, put it in somewhere where it's really, really cold and these white places start to darken up, start to get darker, yeah.
Is that how animals change their fur color? Like
No, you're talking about like Arctic hares and Arctic foxes. I mean, they molt their old fur, but it does play a role in how they deposit pigment in the next set. But they have to like molt their white fur and then replace it with the brown. Yeah. All right, it's 12:40. We're out of time and we're out of slides. It's perfect. Perfect. Have a wonderful day. If you have lab, I'll see you.